Hello everyone, hope you're still safe at home and in your health. Okay, so last week we spoke about the lymphoid organs and the cells of the immune system. This week we're going to talk about how the immune system cells, the lymphocytes in specific, recognize the antigen of the bacteria, for example, or the pathogen. Okay, so let's start with document 6, book page 125. Antigen recognition by B lymphocytes. Remember, we said that B lymphocytes have to do with antibodies. And as we said or mentioned last time, they are formed in the bone marrow and they mature in the bone marrow. So antibodies, or what is known more scientifically as immunoglobulins, Ig, Ig is short for immunoglobulin. So they're present either on the surface of B lymphocytes or they're going to be circulating. This is basically a B lymphocyte, and these are the antibodies, okay? And these are the ones that are going to recognize the antigens of the non-self. So first of all, you need to know that they are Y-shaped, and they are formed of 4 polypeptide chains, 2 heavy or 2 large, and 2 light. They have a constant region and a variable region. Just like you might guess, a constant region is the same, just a little slight variation in it to give me 5 different classes of immunoglobulins: IgM, IgD, IgG, IgA, and IgE. You're not supposed to know what each one of these classes is responsible for. All you need to know that a slight variation in the constant region of the antibody would cause 5 classes of immunoglobulins. Now we go to the variable region, and this is the one that recognizes the specific antigen, and it's different between antibodies. So if you open your book to page 125, this is the diagram of an antibody. Okay, you see we've got the heavy chain, the yellow one, the large portion. This is why it's called the heavy chain. Polypeptide, as you should know by now, that it's a peptide chain, which means it's made of amino acids. Amino acids have to do with proteins. And we've got the light chain, okay? So heavy chain, light chain, Y-shaped. This is the region of variable sequence. These two, the dark ones, and the rest is the constant one with the slight variations to create 5 classes of immunoglobulins. The classes and forms of human immunoglobulins. In number 1, we've got the monomers, or 1. So 1 antibody. This is the IgG, IgD. You see they all have the Y shape. They just have slight variations in the constant region. Over here we've got 3, over here 3, 2, 2. Okay, so slight variation in the constant region. You see the red chain over here? Over here it's not present, it's present here. This is why we've got the 5 different classes. So monomer, which means 1, dimer, which means 2 antibodies connected, and polymer, which means several antibodies connected. Now we come to the important part, or how does the antibody recognize the antigen? So first of all, you need to know that there's a specificity to the antibodies. They recognize soluble or cellular antigens. So the ones present on the surfaces of pathogens are the ones that move around alone in the blood. And the antigen binding site binds to antigenic determinants. So it doesn't bind to the whole antigen, it binds to part of it. This part of the antigen is known as antigenic determinant, or epitope, to make it easier for you. So, and you need to know that the binding site of the antibody and the epitope, they have complementary shapes. I'm going to tell you just how in a minute. And they fit each other just like the lock and key. So if you look over here at document E on book page 126, this is the antigen binding site. So the green one is the antibody, and this is the epitope, or the antigenic determinant. You see how they fit each other? The shape fits, and they bind to each other through a non-covalent bond. Now this is what's really important, the cross reaction, which tells you that an antigen may have several epitopes and can bind many different antibodies However, an antibody is not specific for an antigen, but rather for an epitope. So one antibody will bind to one epitope, not to one antibody. So, not to one antigen. So if I have several antigens having the same epitope, I could create only one antibody to it. And this is the cross reaction. And so you see over here, this is the antibody, the Y-shaped antibody. This is the epitope. Over here it's Z, and over here it's Z. But look at the antigen. Over here I have X and Y, and over here I have U and T. So I have different antigens, but they have the same epitope, so they bind the same antibody. That is, it can bind two different antigens whenever they have a common epitope. We say they cross-react. This is it for document 6. We're going to move to document 7. Antigen recognition by T lymphocytes now. T lymphocytes are done, or they are formed in the bone marrow, travel to the thymus in order to mature. So we have the T lymphocyte receptors, and they are the ones that recognize antigens which are present on HLA of self. So the TCR is supposed to bind to the HLA. This is why, remember at the beginning when we said that they eliminate the TCRs that bind to self-peptide but not to self-HLA? This is the reason behind it, because the TCR binds to the self-HLA and it checks the peptide of it. If it's the self-peptide, it doesn't kill it. But if it's the non-self peptide, it doesn't recognize the peptide, a reaction would occur. So let's start with the molecular structure of TCR. Again, this is formed of two polypeptide chains. The orange one is one chain and the green one is another chain. Two polypeptides chain. We've got a variable region and we've got a constant region. This is the molecular structure of a TCR. You need to know that the TCR does a double recognition. So this is the antigen presenting cell. Okay, so this is our normal cell, the cell that's infected. We've got the HLA, and then we've got a peptide. The HLA is self, while the peptide is non-self. This is what the T cell does. First of all, it recognizes the HLA, and second of all, it recognizes the non-self peptide, thus the name double recognition. We have two different types of HLA. We've already talked about it before. Class 1 and Class 2. Class 1 are coded by loci A, B, and C, and they are present on all the nucleated cells. Class 2 are coded by loci D, P, D, Q, D, R. These are genes present on chromosomes, and they are found basically on some some leukocytes like the macrophages. We're gonna move to book page 128. This is the big picture basically. So we've got 1, 2, 3. You see we've got a TCR. We've got T lymphocytes, sorry. This is TC, cytotoxic T cell, and this is T helper. You've got the pictures divided into orange and blue. There's a reason for that, and you're gonna know in a minute why. Normally, in every cell, samples of self-proteins are fragmented into small peptides. The peptides are attached to HLA class 1 molecules and transported to the cell surface, where they are presented to TC cells. However, there are no mature TC cells specific for self-peptides. So let's see. This is a normal cell nucleus. It does self-proteins. The self-proteins are attached to the HLA class 1. Class 1, yes, class 1. Okay, then we've got the HLA1 peptide complex, which travels to the cell surface. Do we have any TC cell attached to it? No, because just like we said that TC cells are, we do not have mature TC cells specific for the self-peptides. Again, the green ones, the green dots over here are self-peptides. But what happens if I've got a virus taking over the cell? This is what happens. The virus would go in and would produce viral proteins. The viral proteins will later be attached to the HLA class 1 molecules. So we're still in the same cell, the nucleated cell. Will attach to HLA class 1 molecule to create HLA1 peptide complex. But this time we don't have self-peptide, we've got viral peptide. It travels to appear on the surface And I've got TC cells that could bind to non-self peptides. Thus, we've got a TC cell which bound to the HLA class 1 molecules and to the non-self peptide. Once this occurs, it will become activated and a reaction would occur. This is if I'm talking about a nucleated cells, which means HLA1. So what about if I'm talking about a macrophage? When a macrophage phagocytoses and digests cellular or soluble antigen, the resulting antigenic peptides are attached to HLA class 2 molecules. They are transported to the cell surface and presented to T helper cells. The T helper cells that recognize these particular peptides are activated. So this is a macrophage. This is why it's blue. It ate a bacteria. It phagocytosed it. The bacteria was digested and we created bacterial peptides over here. What happened? The HLA class 2 came and took the bacterial peptides and to the surface of the macrophage, where it was represented or presented to T helper cells. Once the T helper recognizes that this could attach to the HLA class 2 and the non-self peptide, it becomes activated and again a reaction occurs. This is it for this week. We're not going to move into a new chapter until next week. So your homework would be probing the documents on book page 126, numbers 1, 3, 4, and 5. It would be on the iSchool. And on book page 128, numbers 2 and 3. Okay, I hope you're all safe at home. Stay home and stay safe. Thank you.